Gulf of Mexico Alliance has a bi-monthly Wednesday webinar series, and Candace reached out to us about co-hosting this webinar on harmful algal blooms because we felt like it would be a huge interest to the National Water Quality Monitoring Council group, and so we're really excited to co-host this webinar together. And since we might have people who are not familiar with the Gulf of Mexico Alliance, I'm going to give a brief introduction to the Alliance, just a few slides before we go into Kate's presentation, to give you a little bit of background about who we are and some of the things that we do. So the Gulf of Mexico Alliance is a nonprofit organization based in the Gulf of Mexico region. It's led by 5 Gulf states with a mission to work together to build partnerships for a healthier Gulf of Mexico. So we work to increase regional collaboration focused on the environmental and economic health of the region. And it's led by the 5 Gulf states with additional partnerships in our network that include 13 federal agencies, academics, businesses, and nonprofit organizations. So we represent a broad partnership of organizations and individuals across the entire region. We have 6 priority issue teams that we focus on across the region, and so these priority issue teams are based on priorities that have been identified by the 5 Gulf states as issues that are common across the region and things that we can work on together to improve. And those include resilience, habitat, wildlife and fisheries, education and engagement, and then of interest to this group, water resources, and we also have data and monitoring. So in addition to our 6 priority issue teams, we also have 3 cross-team initiatives. So these are cross-cutting issues that are of interest to multiple teams, and they include conservation, restoration, resilience, ecosystem services assessment, and marine debris. So these are things that our teams partner on and work together because they're issues that, for example, marine debris is of interest to education and outreach, wildlife and fisheries, water, and so we really have a lot of different folks who have a stake in these issues. And just to give you a real quick overview of how we work, what guides us is our Governor's Action Plan. And so this is our roadmap for the work that we do in GOMA. And Governor's Action Plan, that's where the priority issues are, and then with each of those priority issues, we have specific focus areas and actions. And so we're right at halfway through our action plan right now, and we're well on track with our actions. And for example, related to water resources, there are 3 focus areas: human health, aquatic health, and economic health. And then under each of those focus areas, there are specific actions. And so that's where, for example, Kate's work with harmful algal blooms comes under the umbrella of the work that GOMA is doing. And just, I know that was a very quick overview, but I didn't want to take too much time away from Kate and her presentation. There are some upcoming meetings that I just wanted to put on your radar if you are interested in learning more about GOMA. We have team meetings for each of those priority issue teams. Our resilience team is meeting in just a few weeks in Austin, Texas. And then our data monitoring team, habitat resources, and water resources are having concurrent meetings in January in Baton Rouge, Louisiana. And our education and engagement team is meeting in late January in Biloxi. We also have a monitoring community of practice that will be meeting in January. So those team meetings for data and monitoring, habitat and water resources, and the monitoring community of practice, that will be one larger meeting together over 3 days, where part of it is the monitoring community of practice and part of it is the team meetings. And then In June, we'll have our all-hands meeting, which is where everybody comes together. We typically have about 400 participants from across the region, and we have overall sessions, and then we also have individual team meetings as part of that. And so it's really a good opportunity to come together and work on issues across the region. If you're interested in knowing more about the work that we do, you can visit our website. There's a way to sign up for our newsletter and for updates. We're also on social media, or you can feel free to contact me directly, and I'll be happy to give you more information about the work that we do and any topics of interest that you might like to know more about. So with that, I'm going to turn it over to Kate and let her talk about the work that she's been doing. As part of GOMA, she works with our water resources team as a state lead, and she's also been working on 2 Harmful Algal Bloom projects that were funded by our GOMA Gulf Star program. So that's my last slide, and Kate, you can swap over and take it from here.
Great, thank you.
Thank you, Christina. Kate, I gave you presentation privileges, so you should be able to share your screen. And Christina, we are getting a little bit of feedback from the background, so I'm just going to mute you until the end, if that's okay.
I'm just getting it into slideshow mode. Huh. Okay, so it looks like, let me just escape. I'm gonna blow it out. There's a little bit of an issue with some animations. Okay, let me just fix this really quick. Sorry about this.
We understand.
Otherwise, it's going to keep playing, which is not the intention. So while I'm doing this, I just wanted to mention, so I'm going to give an update on some of the collaborative GOMA projects that we have funded, and I thought that given the request, one of the best ways to do that would be to provide a little bit of background about red tide, and then focus to some extent on the bloom that we've been having this past year. Okay, so we may not get to have that first slide, but that first slide is simply showing a number of different phytoplankton species. And although we have thousands and thousands and thousands of different types of algae or phytoplankton, only a handful of those are actually toxic. And these microscopic phytoplankton are responsible for supporting our food web. And they also are responsible for, they do produce half of the air that we breathe. So we often think about terrestrial environments as being very productive, but the ocean is also very productive in terms of producing oxygen through photosynthesis. So what is red tide? Red tide is commonly referred to as a common term for harmful algal blooms. There are a number of different types of species that are referred to as red tide. Harmful algal blooms are unique in that these are species that can produce toxins. These blooms can form when algae proliferate, and these toxins can cause a number of different issues. When blooms proliferate, they can also result in low dissolved oxygen, and this can have negative impacts on the ecosystem as well as various wildlife. I've shown here some electron micrographs of Carenia brevis, as well as the structure of one of the main forms of brevetoxin. Carenia brevis produces over 20 different forms of brevetoxins, so those forms can vary with respect to their toxicity. On the left, I've shown here those toxins can have impacts on wildlife, and we know that they can kill fish, birds, sea turtles, manatees, as well as dolphins, and we've certainly seen quite a bit of that during this event that we've had ongoing. red tide can also have numerous human health impacts. One way that this can happen is when cells are in the water and then filter feeding organisms like shellfish, clams, oysters, mussels. They can accumulate the toxin, and then they can cause neurotoxic shellfish poisoning in human consumers. And so that's something that we have to be very careful. Karenia brevis actually has a harvest area closure when cells reach 5,000 cells per liter. And it's the only alga that has a closure tied with cell concentrations rather than toxin concentrations in shellfish. The toxin is also able to be carried in an aerosolized form, and that's either via sea spray or cells breaking open near the beach, and that can cause respiratory irritation in humans, and some people are more sensitive than others to that. And so this is showing basically on the right here, we see an area where we have red tide. There's a very clear demarcation where we don't seem to have red tide on the left. And this just highlights one point that I really want to make, which is that it takes a highly integrated and interdisciplinary network to observe, predict, manage, and mitigate red tide, as well as the associated impacts. So I'll try to give a little bit of an overview of that today. First, I wanted to introduce Karenia brevis, the red tide alga. So this is a dinoflagellate. It can swim. Hopefully that video is coming through. It does have a unique swimming behavior. I'll bring that up again later on, but just something to note as you watch these cells move around. So they are able to swim. They are able to vertically migrate in the water column. And we have, as I mentioned, a huge, extensive monitoring and research program. The two go hand in hand. And we work very closely with the Florida Department of Agriculture and Consumer Services, or FDACS, and they're the management authority for shellfish harvest area zones in Florida. Our institute provides technical support. We do harmful algal bloom monitoring and biotoxin testing. We also do a lot of research to support our efforts in examining both cells as well as toxins. And we work very closely with a number of different partners shown kind of throughout the slide, as well as our own different groups at the Fish and Wildlife Research Institute. One of those groups that we work most closely with because of the fish kill impacts that Karenia brevis causes is our Fish and Wildlife Health Group. We also have a very extensive citizen volunteer network, and I'll explain a little bit more about how we get samples. Just a second. So there are a few things that I'm going to talk about today. I'm going to try to make them go together, but I have two projects that I'll be discussing in the midst of explaining some of the present bloom. And one of those projects is focused on predicting Karenia brevis at seasonal timescales using gliders. The other is focused on detecting Karenia brevis using a very sensitive genetic method. And so while they don't necessarily immediately make sense in terms of how they go together, I will be talking about both, and hopefully that'll come together a little bit. So if we take a big step back, there's a lot of interest in being able to predict harmful algal blooms so that we can better prepare for the potential impacts, but also so that we can have a better sense of the mechanisms and drivers underlying these blooms. They change from year to year, and the different drivers underlying the blooms may or may not be the same from event to event. And so to basically look at bloom dynamics over the course of 25 years, we were able to work together, and I'm using the royal we here, to identify some of the most common circulation patterns that are observed in the loop current. And that is the current that I've shown here. You can kind of see it highlighted in red in the Gulf of Mexico. And so the strength and location of this current is very important, we believe, for Karenia brevis, in particular by controlling the flux of the nutrients, but also the direction and the timing of the currents that may deliver cells into the nearshore environment. And so on the right-hand side, Each of these different panels represents one of those most common patterns that have been observed since 1997. And so we can look at these and get a better sense, again, for which conditions seem to promote Corynea brevis. And in an ideal world, we would use this prior to the start of the bloom to help get a sense for how intense and severe the bloom might be later on that season. Oops, we've got some more initiation, or more, let's see if we can keep it there. So we know that blooms initiate 10 to 40 miles offshore at depth in the Gulf of Mexico. We also know that these blooms occur nearly annually in southwest Florida. And this is just a schematic showing how the combination of both surface winds as well as those offshore currents, like the loop current that I mentioned, they play a role in setting the stage for the currents that then are transporting cells onshore. These currents are often actually they're southeastern, that's a typo, but they are often moving to the southeast. Those are bringing cells into the nearshore environment where they can then be upwelled into the surface. Once we have that upwelling into the surface, we often see blooms in coastal areas, and that's typically where we see fish kills and those sorts of reports. We know that, Corynea brevis is ecologically flexible. So in terms of its ability to persist across a broad salinity and temperature range, we know that it can do that based on both lab and field studies. And we also know through a lot of different work that's been done, Corynea brevis can use a diversity of nutrient sources for growth. This schematic is just showing some of those, but that includes both oceanic as well as land-based sources. We know that there are other factors important for phytoplankton growth. If you look over to the right, I've shown the sun there. With the new LED-based technology, we're now able to grow cells in the lab and approximate the conditions that those cells would experience in the very surface of the ocean. And so in terms of looking at the ecology, we're finding that although prior reports may have suggested that Corynea could grow very well at low light, which would make sense given this offshore bloom initiation hypothesis, we're also finding that cells can grow well at high light. So we're adding that to the list of areas where Corynea continues to seem to be ecologically flexible. This is important for thinking about why blooms may persist for so long. Okay, so jumping into the glider program, I wasn't sure how many people knew exactly what a glider was, so I wanted to show a few pictures. They look almost like a torpedo, and they effectively fly missions out in the environment. You can set those missions ahead of time, and they move up and down throughout the water column. They have a number of different sensors that are important for oceanographic measurements, including temperature, salinity, chlorophyll, as well as others. Each day, or as they hit the surface, that information that they've collected is transmitted via satellite back to land. And so we're able to use these to get a sense of what's going on, not just at the surface of the water column, but throughout the water column at depth as well. And so you can see, again, a picture of it before it's deployed, and then a picture of it right as it's going in the water. So we've done several different glider efforts that have been supported by GulfStar. And just to give a sense, we are working with GOMA funding to be able to augment missions that effectively have already been planned. And so this was our first trip that we did with GulfStar funding. There was a deployment that was already planned offshore of Tampa Bay. And those original plans were for a 10 to 14 day mission that approximately went 60 kilometers between the 30 and 55 meter isobaths. And what we were able to do is extend the mission for an extra 10 days, and we also were able to go out much further to the 10 meter isobath. And this was really nice because it allowed us to go offshore much deeper than we normally go. And first I'm showing salinity here. And so you can see that there is some structure, and just to kind of orient people, this is depth along the y-axis, we have time along the x-axis, and then we have the salinity, or whatever the measurement is, shown in color. And so you can follow along. We deployed here, and so that's the start. And then you can see as we go offshore, it gets deeper, and as we go back onshore, it gets shallower. So you can see some chlorophyll, or some salinity structure. That's very interesting to us. One of the things that we're really looking for when we go out there is chlorophyll, and that's because Crania and many other phytoplankton all phytoplankton have chlorophyll, and that signal is something that we can detect with the glider. And so what we're seeing now here is a persistent chlorophyll feature that is found at approximately 40 meters depth. And we did have a second trip out later that year, approximately 3 months later, and we saw that same feature way offshore. And so some of the longest documented blooms in terms of our prior observations going back to 1953. And this is if we consider bloom concentrations to be over 100,000 cells per liter. We are currently in a bloom that has lasted 13 months. We're about to enter the 14th month. I don't think it's going to stop in the next few days. You can see that the longest lasting bloom on record was 30 months. So this bloom began in November of 2017. It's still very much ongoing. I'm going to give a quick time series going through the bloom. And just to give a few bits of information about this, red tides do occur commonly, nearly annually along Florida's Gulf Coast, and we know that fish populations have resolved to be resilient to those impacts. And in fact, there have been several severe red tide events, and even after these events, fisheries are able to rebound. And we also know that physics, as well as other factors, potentially have played a role in the persistence of the bloom that we've had in 2017, 2018. And so first, I just wanted to mention that for 2017 and where we're at right now into 2018, we did not issue seasonal predictions as we intended to through our project. And I should mention that the seasonal prediction project is funded through NOAA, and that's a project that's being led by Bob Weisberg at the University of South Florida. In order to help ground truth those predictions and also to get a better sense for what's going on with the bloom, we rely very heavily on light microscopy. And so you can see this is a picture probably from maybe 4 months ago. Since then, the space has even been changed so that we have another microscope and another computer in there so that we can get more samples processed. So over 13,000 since the event began in November 2017. Now we'll do a quick time series to go through those. These are just showing monthly snapshots. And typically, just to give a sense, I said that this was one of the longest lasting blooms. Typically, we might see a bloom show up in Southwest Florida in late summer, early fall, stick around for several weeks to several months, and then ideally dissipate. So now we'll see 2017 was certainly a little bit different. Just to help orient you, I've put a green sort of star over Sarasota County. You may want to kind of keep an eye on that, and you'll see the bloom moving north and south as we go through. So then, in October, we didn't really have too much going on. November, we started to see in Southwest Florida, especially central Sarasota and south, higher concentrations. Same in December, same in January. But now if you look down to the south, we can see that in Monroe County, we're starting to see some impacts. And that was due to southern transport of the bloom into Florida Bay, where it caused fish kills. That occurred yet again in February, and we had that same sort of pattern. So now into March, we're seeing that the bloom has moved back north to some extent. In April, it's contracted a little bit again. In May, it moved north a bit. And in June, it expanded a bit, and it's moving north again. In July, we had a lot of areas that had high cell concentrations. And then in August, we see that the bloom moves north to Pinellas County. So you can see it expanding north. And then in September, we see the bloom expand to both the Panhandle as well as Florida's East Coast. And while it's fairly common to have blooms on the Panhandle, it doesn't happen every year, but it happens every 2 or 3 or 4 years or so. We've only had a handful of prior events on the East Coast that are related to transport of the Southwest Florida bloom along south of the Keys and then up the East Coast. The prior event where that happened was in 2007, and those cells were actually transported up the East Coast of the United States. So I'll pause here a little bit. I just wanted to give a sense from September. There were a few things that we've been trying to do weekly to biweekly aerial surveys during this event. For those of you that haven't seen a bloom, it is very visible. So I just wanted to give a sense of what the bloom might look like. This is a bloom that's quite near shore. We definitely saw concentrated cells along fronts offshore. And then we also saw patchy red tide, and you can almost see these finger-like tendrils, and those were 7 to 10 miles offshore. Oh, let's see if we can get that. One other interesting thing that we saw from this survey and a number of other surveys was the presence of a different alga, Trichodesmium. You can see that alga in the golden brown color here. Again, it seems to concentrate along these sort of gradients and transition zones. You can see that Carenia brevis is based on the water discoloration in relatively close association. This type of alga, Trichodesmium, is particularly interesting because it is able to fix nitrogen, and it's thought that iron dust from the Sahara helps relieve iron stress and allows Trichodesmium to bloom. Because Trichodesmium can fix nitrogen, it can remove nitrogen or use nitrogen from the atmosphere, and it can convert it into different forms that Carenia can use, like ammonium or dissolved organic nitrogen. And 2018 was a particularly bad year for dust. This is showing an image from, I believe, June from NASA, and you can see that dust making its way from the Sahara across into the Gulf of Mexico. And there were events in the eastern Gulf of Mexico as well as the western Gulf. So in this same time period, starting in late August and then going into September, we had yet another collaborative glider effort. And so this is showing the general trajectory. This was off of Tampa Bay. And so you can see that it was deployed offshore, and then it went offshore, and then onshore, and then kind of went north, and then south, and then back onshore. And so with that mission, that explains now where when we look over time, we're seeing it kind of move offshore, inshore, offshore, inshore. And what you may see, and if you remember back to the prior glider data that I was showing you, we're still seeing this subsurface chlorophyll peak. And so we were interested in trying to get out and sample that. And we've, in fact, found, I don't know if you guys can see this, but we did have 9,000 cells of Carenia brevis per liter in that elevated chlorophyll area when we were able to go retrieve the glider. However, that was somewhat complicated by the satellite data. So now this is showing sea surface chlorophyll data. This is a product that is put out online by the University of South Florida's Ocean Optics Group. And so this, we're having sea surface chlorophyll with the warmer colors in red, showing higher chlorophyll levels. And again, we can't confirm that that's a Corynea brevis bloom without going out and collecting a water sample, but given what has been going on and our monitoring results, which are shown here in the little dots, and again, those warmer colors indicate higher cell counts. I should have mentioned that earlier. So if we look to where this glider was actually retrieved way offshore, we can see that not only did we have Corynea brevis observed at the surface as well as at the bottom, but you can also see that there is a surface feature of chlorophyll that seems like it was transported offshore from near shore. And so this, I think, is a great illustration of some of the complexity underlying these bloom dynamics, but also how some of the tools that we use can be integrated to look more comprehensively at the system and get a better sense for what might be going on. It also hopefully illustrates why predicting what was going to happen with the bloom in 2018-2019 was a challenge. Okay, so then in October, I wanted to include this before we show current conditions because October was a pretty rough month in terms of cells. You can see that now we have a pretty full-fledged bloom all along the East Coast up until about Brevard County. And again, that bloom was transported south and around the Keys, so we know that the southwest bloom was involved. In terms of the northwest bloom, that's still more of a question. We know that there is likely one initiation zone for Corynea brevis that feeds both southwest and northwest Florida. And so there's still some question as to whether surface populations were transported via some of the storms that we had go through, and or whether the northwest bloom was introduced via some new cells introduced at depth. So that's something that we're still trying to look at in more detail. Okay, so then we have slightly better news. This was last week's status report. We are still unfortunately seeing a pretty bad bloom in southwest Florida. If you look to the East Coast, conditions, knock on wood, have improved there somewhat, and we're still seeing concentrations, bloom concentrations in the Panhandle, and we've had reports from some other Gulf states that they have observed Corynea and or potential impacts. And so again, this is our most recent map. That satellite data kind of gives us a sense of what's going on at the surface, and we can also use our data in combination with physical models to issue more short-term models, or short-term forecasts, excuse me. So these are forecasts that we're already issuing. They are separate but connected to the long-term seasonal forecasts that we would like to issue in the future. And these are 4.5-day forecasts, and they show surface currents as well as near bottom currents. And I just wanted to show these because throughout the course of the bloom over the past several months, we have had this north-south movement several times, and we've also had this sort of expansion to other areas. At the present time, we've had persistent south-southern currents, and that's been happening for the past 2 weeks or so. And you can see now with these, so these maps are showing surface on the left. The little X's indicate either where a sample was collected or where a drifter that simulates a sample was dropped into the model. And then you can follow to see where that's likely to be at the end of 4.5 days. And so even though we've had this persistent southern flow, you can see that at the end, near the end of this path, we are expecting to have a reversal in the currents. And whether or not that contributes to offshore push or perhaps that same retention that we've seen in the system, that's something that these types of predictions are very valuable for. I think it also helps support the need for longer term predictions beyond the 4.5 days. And we're continuing to keep an eye on what's going on near bottom because that could be bringing new cells into the system. So again, we're trying to keep an eye on both areas. Okay, so some of the other ways, so I mentioned we've had 13,000 samples collected and processed via light microscopy since last November. And these data are transferred daily to our partners like USF to produce the tools that I've been showing you, but they're also transferred to NOAA to help put out respiratory irritation forecasts. And this is showing the current respiratory irritation forecast, or at least as of yesterday. And you can see that this is based on a combination of factors, not just cell counts, but winds, currents, models, as well as other data sources. So this is another way that we're using a lot of this information to help predict the bloom and its impacts, where it might end up. One of the ways, and this is a project that I'm peripherally involved with, but there has been an effort led largely by GCUS and NOAA to help get something called the HABscope up and going. And this isn't a part of the GOMA-funded project necessarily, but I wanted to mention it because it's a new technology that we've been using in tandem often with some of the other methods. And at the beginning, I showed you that video of Crania swimming. And the HABscope is a simple microscope. It has a 3D printed arm. That arm you can put in an iPod or an iPhone, and basically you take a 30-second video of a water sample. And through uploading that video to GCUS, the Gulf of Mexico Coastal Ocean Observing Systems website, that video is then able to be processed using something akin to facial recognition software that's able to pick out the Crania brevis swimming cells and track them so that they don't get counted more than once and come up with a cell count. That whole process takes about 5 minutes. And so as we continue to need more observations to help get a better sense of where blooms are occurring, this is a method that is something very amenable to citizen science. Okay, so, and then I wanted to use that to transition to talking a little bit more about the Genetic Probes project. So this is another NOAA-funded effort, and there is a probe that's been designed that is specific to Crania brevis. What I did mention at the beginning of my talk is that there are other Crania species that live in the Gulf of Mexico, and Crania brevis is morphologically similar to those. I've shown just a few of those here. I can also say from personal experience, Crania brevis has a huge range of morphological variability itself, and so that's another factor that we have to keep in mind, especially when we have a bloom where we may have some cells that are more or less healthy than others. Of these 12 Crania species recognized globally, approximately 6 are known to occur in the Gulf. And these species, as I mentioned, can sometimes be difficult to identify. And based on our current screening method, our limit of detection is 333 cells per liter. So we are missing a few orders of magnitude. And this technique is something that is very good at detecting low levels of Crania brevis. It's also effective for detecting a range of cell concentrations. So this is called an amplifier, shown here in the middle, and it basically is an in situ device that does isothermal PCR amplification or isothermal amplification through NASBA, and NASBA stands for nucleic acid sequence-based amplification. John Paul is the lead on the USF project for this in situ detection, and his lab developed an assay that can recognize Crania brevis, and it's been modified several times so that it is appropriate for this in situ detection method. And basically through the course of the project, we have come up with a simplified way to filter seawater, and we basically do that using a spin column that you might normally use for DNA or RNA extraction, and a hand pump. So that's very simple and affordable, and it's actually very quick as well. This allows us to lyse cells and go through a modified RNA extraction. We can then conduct the NASBA out in the field and do this isothermal amplification. And we run all of our samples with a control so that we have a sense. And you can see these two samples that we ran. The top one, K. brevis, is absent. I'm sorry, there's not more color differentiation between those two, but the slightly darker color is Cornea brevis. So you can see that our control did amplify. And the bottom one, Cornea brevis, was present. And if you look, we started to see that positive signal at approximately 10 minutes after the instrument started to run. So for this project, there have been, one of the goals of the project is to try to turn this technology over to end users, and we are one of those end users. Hold on just one sec. And so one of the first goals of the project was to basically transfer that technology to the state of Florida so that we are able to use it. And that's been really successful, and it's something that we've been using in our group. And again, that low-level detection is something that is really great for us because it helps us get lower concentrations than our current microscopy methods. So that was our first goal. We've also been working really hard to engage a number of different partners in Florida as well as Gulf-wide, and GOMA has been immensely valuable for that and has given us insight into some of the different HAB issues that might need this sort of assistance in other states. Pseudonychia is one, Alexandrium is another, and so that's been really beneficial. We also are interested in trying to transfer this technology Gulf-wide, and so we've gone through a number of different trainings to help determine what the best way to do that might be. So these are some of the areas where we've had those trainings shown here on the map. And the name of the project is actually the Cornea tricorder. So for any Star Trek fans out there, you may be aware that that's sort of an environmental sensor, and so that explains the appearance of this critter in the Apalachicola training. We've also worked closely with the St. Petersburg College and done a lot of training with students there, and have had several students who have done theses that have focused on using this particular technology. It works quite well. This is showing a comparison, and this is from a number of our field trials as well as volunteer trainings. And so what we have on the x-axis is our light microscopy, cells per liter, and then on the y-axis we have our genetic quantification via NASBA. And so what we can see with this is that we do generally have a slight overestimation of the cell counts when we compare NASBA versus light microscopy. But if we look at the correlation between the two, there's a good, strong correspondence. And so we have done additional experiments in the lab to better examine that difference and what might be going on there. I'm not going to go into that today, but I did want to mention some cool updates that have happened on the project. So once we generate this data, we are now able to upload it to the GCROS website, to their HABscope website, and we have our own separate RT NASBA little tab on there. And so users can sign in, and I'm just going to skip through these pretty quickly. It will tell you if you have a good sample upload, And then this is the information that you get out of that. And again, I won't go into too much detail, but basically you get your data in a number of different formats that have walked through the different analysis needed to relate the fluorescent signal that we get through the NASBA assay back to cells per liter. And so we have just several steps, again, that GCRUS is automated so that we can get to this final answer of cells per liter. So that's been a huge step forward. Just to give an overview of the assay, the prep time can be 30 minutes or less. The run time is approximately 60 minutes, and then that data upload is very quick, less than 5 minutes. The amplifier itself costs around $9,000, and our estimate per sample is approximately $25, and that includes a lot of the consumables as well as the buffers and reagents. And I mentioned that it has a great limit of detection, and that's approximately 1 cell per reaction or 40 cells per liter. And we've used this ourselves a few times out in the field, and we've taken it out on research vessels in areas where we knew red tide was close by, but not in that particular area, and we did detect what seems to be red tide right at that very low level, near the limit of detection even of this method. And again, that's very interested for thinking about bloom initiation and where cells may be residing in the water column or possibly even other areas. So this is going on right now as we speak, but this is this week's volunteer sampling array. So we have someone in our group, Alicia Hoagland, who's been working on this project, and she has been very energetic in terms of reaching out and trying to get Gulf-wide coverage, or at least up to Louisiana, and to try to, again, get a snapshot of what's going on and to get samples that we can then use for additional analyses to help better resolve some of the potential issues or needs that we may have in working with Karenia Gulf-wide. Some of those I think I've previously alluded to, but we've worked a lot with cultures in the lab, and we do know that there is some variability in terms of their expression of the gene as well as the copies of the gene that they may have. And so that's really important to understand for thinking about how we convert that genetic signal back to a cells per liter signal that's tied to microscopy data. So, so that's something that's going on this week, and we are very grateful. I don't know if we have anyone from Alabama and Mississippi, our GOMA team members on the call, but they've been a great help in terms of getting access to those areas. So before I take any questions, I want to make sure to thank these key partners that were involved in these particular slides and the GOMA projects that I've been showing. And I also want to kind of go back to that original point where in terms of thinking about the best way to predict Karenia, understand what's going on, I think that strengthening our capacity to make observations quickly, easily, cheaply, using a variety of different formats is going to help us in driving that prediction to the next level. And so with that, I'll stop.